This is an overview of the structure of TPC1 determined by X-ray crystallography. Two-pore channels, or TPCs, comprise a subfamily of eukaryotic voltage-gated and ligand-gated cation channels with two non-equivalent tandem pore-forming subunits that come together to form quasi-tetrameric channels. Found in vacuolar or endolysosomal membranes in the cell, they integrate changes in membrane potential, second messenger molecules, and phosphorylation to regulate cellular ion homeostasis, intravesicular pH, excitability, and endolysosomal trafficking. TPCs have been linked to numerous diseases such as Ebola virus infections, diabetes, liver disease, and Parkinson's disease. Research shows that TPC-specific drug, trans-NED19, can ameliorate the symptoms of these diseases and even provide a cure of 50% of Ebola virus infections in animals. We recently solved the structure of TPC1 from Arabidopsis thaliana bound to drug NED19 to 2.87 angstrom resolution by X-ray crystallography. In addition to serving as a benchmark for understanding the mechanisms underlying voltage sensing, activation, and phosphoregulation of TPC channels, the structure suggests therapeutic mechanisms for targeting these channels in diseases dependent on their action. Plant TPC1 is a non-selective channel. The pore mouth brings 4 negative charges to its lining, which could interact with passing ions. However, the site is too wide to select ions. Below this lies the selectivity filter, which is largely hydrophobic, consistent with the lack of selectivity. TPC1 consists of two non-identical shaker-like pore-forming subunits, domain 1 in red and domain 2 in blue, separated by an EF hand domain in green. Each subunit contains 12 transmembrane helices. Two central pore domains couple the voltage sensing domains VSD1 and 2 and modulatory cytosolic N and C terminal domains and the EF hand domain, which extend 20 angstroms into the cytoplasm. The cytosolic EF hand domain, positioned between domain 1 and 2, facilitates activation by cytosolic calcium. One of the two voltage sensing domains, VSD2, encodes voltage sensitivity and an inhibition by luminal calcium and adopts a conformation distinct from the activated state observed in structures of other voltage-gated ion channels. The central cavity of the pore is filled with water, ions, and solvent. Two rings of hydrophobic residues seal the cavity from the cytoplasm, forming part of the extensively studied pore gate. This supports the idea that the voltage sensors, selectivity filter, and gate undergo coordinated movements during channel opening and closing to regulate ion conductance. The structure shows that the drug trans-NED19 acts allosterically by clamping the pore domains to VSD2. In animals, NED19 prevents infection by Ebola virus and other filoviruses, presumably by altering their fusion with the endolysosome and delivery of their genetic material into the cytoplasm. VSD2 contains a classic voltage sensing motif, R1 through R3, in helix S10. VSD2 adopts a distinct conformation with respect to other voltage-gated ion channel structures, thought to represent the resting state of a voltage sensor. Luminal calcium inhibits plant TPC1 conductance. We observe two calcium binding sites on the luminal side of VSD2. Site 1 does not affect the channel, whereas Site 2, composed of residues in VSD2 and the pore domain, inhibits the channel by shifting the voltage dependence to more positive voltages. A lipid is seen near this site and could play a role in the known regulation of plant TPCs by fatty acids. Two EF hand domains positioned between domains 1 and 2 facilitate activation of plant TPC1 by cytosolic calcium ions. These domains consist of two calcium binding motifs, EF1 and 2. EF1 is purely structural, whereas EF2 facilitates channel activation by cytosolic calcium. We find calcium-bound EF2 forms a complex with the C-terminal domain in orange, suggesting it too may be coupled to calcium activation. TPCs are phosphorylation-gated channels. We determine sites of phosphorylation in the N and C-terminal domains that are positioned to allosterically modulate cytoplasmic calcium activation, shown here as capital Ps. The channel conductance of human and plant TPCs is multimodal, and our structure suggests a mechanism for channel opening whereby calcium concentrations, voltage, and phosphorregulation are integrated to govern the conduction of ions. Further details of this discovery can be found in publications by Kintzer and Stroud and by Guo and Zhang in the March 10th issue of Nature, 2016.